So this is the, I'm going to go through the required practical 2. I'm just going to talk through the method, anything you should pick out or is it of import to know, and go through the questions as well. The mark scheme is right at the back. So this is how you can use a microscope to look at a cell or a tissue and identify if cells are undergoing mitosis or not. Just looking at the method, some of the things to point out. First of all, you have to have a stain that will stain the chromosomes, stain the DNA, and it makes the chromosomes visible. The pterygium blue will stain the chromosomes dark blue. It's really important to use the tip. Plants are quite easy to use because the chromosomes are quite large. So root tip, which is actually the meristematic region, so is the meristem of the plant, so meristematic region, and they're parts of the plant that is undergoing cell division. You couldn't study mitosis in a part of the plant that wasn't undergoing cell division, otherwise the vast majority of the cells would be in interphase and you can't see visible chromosomes. So you want to be able to see the chromosomes being visible after they're coiled up and condensed. So you have to use an actively growing part of the plant, and that's why we use the tip of a root, so where the meristem is, because the cells are undergoing cell division. Just let's look through all of this. So just going through some points in the method. First of all, you have to place in, it's really quite concentrated hydrochloric acid, so it's a strong acid, concentrating, it helps to break down the cell wall. You have to be able to get the dye, the stain inside the cells. Other bits of the method, so you have to cut off you just want the very tip, and that's where the cells are dividing. And the word macerate is a nice word, and it's used to spread out the cells. You don't want overlapping cells. You want one single layer of cells that's very thin, which is why you press down with a cover slip to spread out the cells. So one layer of cells that's thin, so the light can penetrate through, allows it to be visible. You then might be asked to calculate a mitotic index. There are a couple of different ways that you can do this. It's, in essence, you're counting the number of cells in mitosis, and you need to count the total number of cells, and how you represent it can be in different ways. So it could be represented as a fraction, so 2 cells in mitosis to 10 cells in total. This could be represented as a decimal, in which case it would be 0.2, so 0.2. Or it could be represented as a mitotic index as a percentage, in which case you times this number by 100, so 2 divided by 10 times 100 is 20%, so 20% of the cells are in mitosis. Or it could be asked to do in a ratio, which would be 2 to 10. Because you're dividing by the total number of cells, then you divide by 10 in this instance, so it would be 0.2 to 1, where 1 being the total number of cells. I'm not going to go through all of the questions, the mark scheme at the end. I'll just talk through some of the bits. You can see we must use section D because the tip of the root is where the meristem is, where the cells are dividing. The next question here, this is quite an interesting question. Why the cells remain in metaphase? So if you've got your chromatids lining up along the equator, you wouldn't have any spindle to contract to pull the chromatids apart. So the centromere would stay intact. The centromere would not be able to split if the spindle weren't able to contract. Really common how science works question here, how a control should be treated. It's the same in any situation you're using a control. So it should be treated exactly the same, but instead of using caffeine, you've got to use something else instead. So no caffeine, and a good thing to use would be distilled water instead of caffeine. That allows you, why would you use it? It allows you to prove that it's the caffeine having the effect. Now that's not a mark point here, but it's also a follow-up question. It allows you to prove that it's the caffeine having the effect. This last question here is a whole A-level question. Don't worry too much about that one if it's just the AS exam you're revising for. Just looking at the different types of cell we've got here, just run through why they are the different stages. So this must be interphase because there is a clear, distinct nucleus, and you cannot see distinct chromosomes. You cannot see the chromosomes; they're not visible. You just see kind of a haze, a fuzz, kind of like static inside the nucleus. You can also see a nucleolus present, so no distinct visible chromosomes, a nucleus and a nucleolus. Stage A, I think this is quite a tricky, it is metaphase, but it's quite tricky. You can sort of see a line there that the centromeres are on, and you can see the chromatids kind of radiating out from that equator of the cell, from the middle of the cell. So that would be metaphase. Obviously anaphase this, you can see the chromatids have been separated and are moving towards each opposite pole. Another really common how science works question: why did you choose to examine 200 cells? It's a similar question to why in a drugs trial might use 10,000 people. It ensures that it's a representative sample, so that you have definitely got some cells that are undergoing mitosis as well as cells that aren't in there. Looking at this maths question, if you've got 90% of the cells in interphase, that must mean that 10% of the cells are in mitosis, because they're the two general stages of the cell cycle. You can subdivide each, but 90% in interphase or 10% in mitosis. If you want to find out the percentage of something, so in this case we want to work out 10% of 20 hours, very straightforward way to do that: convert the percentage into a decimal, which would be 0.1, then times it by what you want to find. So you want to find 10% of 20 hours, which comes out at 2 hours. Here's the mark scheme over the next couple of slides, so I'll just run through it. Right, that's the first two questions. This is the last question, and it's all in there.